So can one of you please confirm whether my screen is visible to you or not? So we can continue.
Visible, sir. Visible.
Okay, thanks. It's visible. So a very good evening to all. So today we'll be taking some more problems and solving them for organic chemistry in biology and drug development. So let's start from the problem number one. So which of the following will form this palindromic sequence? So here a few sequence has been given. So it is asking which will form this palindromic sequences. So what is the answer? Yes, Trupti and Subhas. So first of all, you need to know what is palindromic sequence. So what is the palindromic sequence? If you don't know, we'll tell. Okay, so palindromic sequence are referred to as the sequence of nucleotides in DNA duplex or RNA, where the sequence in one strand is the same as the complementary sequence of the other strand when read from the same direction on both strands, either 5 prime to 3 prime or 3 prime to 5 prime. So once you read whether 3 prime to 5 prime, 5 prime to 3 prime, you will get the same sequence. Now you tell which one is palindromic sequence. If the definition is clear to you, then you can easily tell which will be the palindromic sequence. Okay, so let me give the example. So correct option is D. The complementary sequence of this one, you need to write complementary sequence of every right. So for this it will be T, and if you see for this option D, so it will be T for A to T, C to G, G to C, T to T to A to T. So this is complementary sequence. So once you read from 3 prime to 5 prime, you have the same sequence. Like if you read from 3 prime to 5 prime, you have a T A A C G T T A, and in the complementary sequence, if I also read from the same direction 3 prime to 5 prime, then you have a T A A C G T T A. So this is a palindromic sequence. Fine. So like if sequence is given, you take the complementary sequence of that and read from the same direction. You should get the same sequence. Those sequence will be palindromic sequence. So complementary of this is this, and you are reading from 3 prime to 5 prime. You are getting TAA, and here also 3 prime to 5 prime. It is TAA, CG, TTA. The same sequence. Those sequences are called palindromic sequence. Okay. Okay, next question. Next question says restriction enzyme EcoRB cleave the double strand viral DNA but does not affect the bacterial host DNA with same nucleotide sequence. Which of the following regions account for this phenomenon? So first option is the rate of cleaving the host polynucleotide is clinically very slow to have any practical effect. Host nucleic acid is methylated and hence no longer recognized by the restriction enzyme. Option C, binding of the restriction enzyme with the viral restriction site is thermodynamically more facile. Option D, host nucleic acid is also cleaved but immediately annealed and hence no change is noticed. So, which option you seems is correct uh, for this restriction enzyme that it cleaves the double strand viral DNA but does not affect the bacterial host DNA. So, what could be the possible reason in these cases? So, in these cases, in bacterial host DNA, methylase enzyme methylates the adenine and that prevents the enzyme EcoRV, this is restriction enzyme, from binding it to the, and hence no cleavage of host DNA occurs. So in this process, this host DNA to the host DNA adenine methylated, so it will not get recognized and hence it will not get cleaved off. So this way it and this viral DNA is not methylated and hence it easily acts as a substrate for the given restriction enzyme. So option B is correct in this case. So if you see here, this restriction enzyme form highly specific DNA cleavages reactions. So if it may happen that you have the same kind of sequence in viral DNA and host DNA also. So, but how this enzyme restriction enzyme will identify that whether it is from host DNA or viral DNA? So, what it does in host DNA, this methylase enzyme, it will methylate this adenine. I mean, amine of adenine, amino group of adenine. Then it will not recognize by this texture enzyme. So it will not cut from the it will not cut to the host DNA. It will cut only leave only viral DNA. This is how it identify. So the host DNA is protected by other enzymes called methylase, which methylate adenine base within host recognition sequence. And for each restriction enzyme, corresponding methylase used to be present. Okay, so here we have a next question. If you have any doubt, please raise your hand, or you can speak, or you can put up your questions in chat box. Consider the following this palindromic nucleotide sequence with the restriction sites indicated by vertical lines. Okay, so there are four options, and their palindromic sequence means complementary sequence. That if you read from same side, you will get same sequence. So and but their different restriction sites have been given. So what it is asking that first and second represent blunt cut, and they are ligated easily with respect to sticky cut. Third represent a blunt cut, and its ligation is difficult with respect to sticky cut. Fourth represent a sticky cut, and its ligation is difficult with respect to blunt cut. First and second represent sticky cut, and their ligation is difficult with respect to blunt cut. Okay, then first we need to know what is blunt cut, what is sticky cut. So any idea what is blunt cut, what is sticky cut? Okay, so in sticky ends, one strand is longer than other, typically by at least a few nucleotides, such that longer strand has bases which are left unpaired. In blunt ends, both strands are equal length, so um, they end at the same base position, leaving no unpaired base on their strand. So here the correct option is B. But the B option is okay. So B option is third represents a blunt cut. Okay, and its ligation is difficult with respect to sticky cut. So here it is saying that third is a blunt cut. Yes, according to the definition of blunt cut, it will cut both strands on of equal length. So this is a blunt cut, and hence it is a blunt cut. So their ligation is much more difficult with respect to sticky cut. Okay, so option B is correct. In all the cases, you have a sticky cut. In first case, you have a sticky cut. First and second cases, you have a sticky cuts with 5 prime ends, right? And in fourth, we have a cuts with a 3 prime this protecting ends. Only in third, we have a blunt cuts. Okay, so next question. Next question is the consider the following diagram related to recombinant DNA technology and identify the correct statement. Okay, so here they have given some schematic diagram which represent the DNA technology. So and they have labeled the steps with the A, B, C, D, E, F. You need to identify with what to correspond to what. So option A is A represent source for the gene to be amplified. A source to the gene for amplified. B represent plasmid DNA from bacteria. Okay. B contains genes expressing protein responsible for antibiotic resistance. C represent exonucleases, restriction enzymes. D represent gene to be amplified. E represents plasmid vector. F represents recombinant DNA. So you need to find what represents what. So in these cases, if you see, A represents the chromosomal DNA which contains the target to be amplified. So A is a chromosomal DNA which contains target gene that will be amplified. And B represent plasmid DNA from bacteria that are highly replicable and contain the genes which express the protein that confer the antibiotic resistance to the bacteria. Fine. So source gene to amplified. B represents plasmid DNA. So in this option, this is also correct. This is also correct. Okay. The same endonuclease restriction enzyme C is used to chop out the target gene. So endonuclease restriction enzyme. C is a endonuclease restriction enzyme. So here C represents exonuclease restriction enzyme. No. So C is not a exonuclease restriction enzyme. C is a endonuclease restriction enzyme. It is so this option is removed. Okay. So A amplifies plasmid DNA from bacteria. This option is also removed. A represents recombinant DNA. B represents source of the gene. That is also gone. So option A represents source for the gene to be amplified. B represents plasmid DNA from the bacteria. B contains genes expressing protein responsible for antibody response. C represents endonuclease restriction enzymes. D represents gene to be amplified. So here D, what D represents, uh, and D the plasmid vector E. The same enzyme endonuclease restriction enzyme C is used to chop out the target gene. So D represents the gene to be amplified, E represents plasmid vector, and F represents recombinant DNA. So this option is correct. So according to this schematic diagram of recombinant DNA technology, option D is correct. Okay. So let's move on to the next question. Is it clear? Okay. So let's go to the next question. Here the question is identify the incorrect statement from the following. So here we need to identify what is the incorrect statement. Option A is between PCR and recombinant DNA technology. Only the latter may be used to express the protein of interest. Okay. So here they are saying that PCR you can amplify DNA from both PCR and DNA technology, recombinant DNA technology, but you can only express protein of interest by using recombinant DNA technology. Yes, this is true. The primary reason for using tag polymerase in PCR it is that it follow a very fast reaction kinetics with DNA primers. Okay. Option C is PCR can be applied upon single strand DNA as well. Option D between DNA polymerase and RNA polymerase. Only the farmer needs primers to initiate the process. So the option B is correct. Tag polymerase isolated from thermus this Acutis. So it's used in PCR. It is thermally stable polymerase which does not get denatured at high temperature range of around 70 to 80 degrees. and shows the optimum catalytic activity at 72 degree. So option C is also correct statement. Option D is also correct statement. However, option B is not a correct statement. So the primary reason for tag polymerases to use, it's not that it has a very fast kinetics. The primary reason is that it is stable, thermally stable polymerase. So because in this process we need to heat to the higher temperature, so we should use the polymerases which are stable at that temperature for denaturation. Okay, let's go to the next question. How many double strand DNA are obtained from the following cases using PCR? So here's the question for the PCR. Okay, three cases are there. In case A, entire double stranded DNA after 6 cycles. Case B has a entire single strand DNA after 7 cycles. And case C is a selected portion of the double stranded DNA after 6 cycles. So it is asking in all the cases how many number of double stranded DNA are obtained. So how to calculate? Okay, let's see. So in case A, when the entire double strand DNA is to be copied, so in case A it is saying this, you need to copy this entire double strand DNA after 6 cycles. So in each cycle, the entire double strand DNA will be copied. So the number of copies obtained is equal to 2 to the power n, whereas n is the number of cycle. So 2 to the power n, how many number of cycles are they? It is asking after. So it is asking after number of 6 cycles. So what will be the correct answer for this? Any one of you? It will be 2 to the power 6, right? Okay, so 2 to the power 6 is 64. So in case B, when the entire single strand DNA is to be copied, it is asking entire single strand DNA after 7 cycles. So number of copies obtained is equal to 2 to the power n minus 1. N is a number of cycles. So 2 to the power n minus 1 means 2 to the power 7 minus 1. That equal to 64. In case C, when only a selected portion of double stranded DNA is to be copied. Here it is asking a selected portion of DNA, double stranded DNA after 6 cycles. Then the number of copies obtained will be 2 to the power n minus 2. So 2 to the power n, 2 to the power n minus 1, 2 to the power n minus 2. So here we have a 2 to the power 6 minus 2. Number of cycles are 6. So 2 to the power 6 minus 2 means 2 to the power 4. That means 16. So which option is correct? So option B. In case A, 64 copies of double strand DNA from the entire double strand DNA, and case B, 64 copies of double strand DNA from the entire single strand DNA. Case 6, 60 copy of double strand DNA from the selected portion of the double strand DNA. Okay, so option B is correct. All right. So next question. Now it is asking identify the correct sequence of processes occurring in one cycle of PCR. So in the PCR, what are the steps involved? It is asking for what are the steps involved in the polymerase chain reaction. Any answer?
C, sir. C. Option
Uh,
number C.
can you please little be loud?
Option number C.
Option number C. Okay. Yes. So option number C. So first is a DNA denaturation at 95 degrees centigrade, then annealing with primers at 45 to 60 degrees centigrade, then extension of the polynucleotide strand by DNA polymerase at 72 degrees centigrade. Yes, option C is correct. So to begin with, DNA melting at around 75, 95 degrees centigrade is carried out to separate the two strand of double strand DNA. Then DNA primers are attached them by cooling the system down to 45 to 60 degree. Then finally the tag polymerase is used for extending the chain at around 72 because this tag polymerase has optimum catalytic efficiency at 72 degree. Okay, so option C is correct. Right. Okay, so let's see next question. So cofactors, types of cofactors. There are two types of cofactors. One, some are metal ions, other are coenzymes. Coenzymes, organic cofactors. Okay, that this I will tell you later. Question is consider the following compounds and identify the correct statement from the following. So this PQRS. They are few compounds, organic compounds, and it is saying Q and R are not pro coenzymes. Only S is water soluble. Option D says only S is a pro coenzyme and water soluble in nature. Okay. P and R are pro coenzymes. All the molecules PQRS are lipid soluble. All the molecules PQRS are pro coenzymes. Only P and S are water soluble. So, what is the answer here? So, let's see. So, coenzymes are usually derived from vitamins. So, there are A, D, E, and K. These are lipid or fat soluble vitamins. That means coenzymes. They are hydrophobic compounds. absorb high, absorbed efficiently with lipids and transported, more likely to accumulate in the body, more likely to lead this hypervitaminosis. And water soluble vitamins are vitamin B and vitamin C. So now, do you know which out of them which one is vitamin C, vitamin D, and A, E, K? We need to identify. Do you know the structure of these? So what it is? What is P? Okay. So vitamin P is a 11 trans retinol. 11 trans retinol means vitamin A. Q is a tocopherol. So P is a vitamin A. Q is a vitamin E. R is a vitamin K1. S is a thiamine. That's mean vitamin B1. PQRS. This is A. What is Q? Q is E. This is vitamin E. S is B1. R is K1. This is B1. This is K1. Okay. So which option is correct? Here we have told that what are the water solubles and lipid solubles vitamins. A, D, E, K are vitamins that are lipid or fat soluble. B and C are water soluble. So let's go to the option. So here it is saying that S is water soluble. That is B1, and here we know that vitamin B is water soluble. Fine. Only S, not Q. And option B is S is a procoenzyme water soluble in nature. So it is not a procoenzyme. P and R are procoenzyme all molecules. PQRS PQRS are lipid soluble. So B A E K B. However, B is water soluble. So this is also incorrect. So option A is correct. Okay. So next question: which of the following mechanistic pathway may be followed as a key step during retinal isomerase mediated conversion of trans-retinal to cis-retinal? So here is the structure of retinal. This is vitamin A. So what will be the key step of mechanism? So it is from the organic chemistry perspective question. Is asking which mechanism it involves. SN2, photochemical electrocyclic ring opening, Michael addition, SN2 prime. So any answer? So here if we see the mechanism, so you have a transretinal. So once nucleophile of the enzyme will attack at this 11th position, this ester group will leave, and this is a SN2 prime reaction which attacks at C11 position. Once you have this SN2 prime at the nucleophilic attack at the 11th position, then there will be bond rotation because now it's a sp3, C11 and C12, and then it will uh, orient the orbitals so that water can attack and this nucleophile can leave again, leading to the cis retinol. So this is how this isomerization happens in case of this retinal. Okay, so the first of D option is correct here. So vitamin A and vision. Vitamin A is necessary to form rhodopsin and adopsin. These are visual pigments. Retinaldehyde is a prosthetic group of light sensitive opsin protection. Okay. So what are the other metabolic function of vitamin A? So it for sure contribute to the vision. Apart from that, it also involved in gene transcriptions, immune functions, embryonic development and reproduction, bone metabolism, hematopoiesis, skin health, and antioxidant activity. So deficiency of vitamin A can cause several diseases. Okay, so another question is what of the following reaction is involved in photochemical conversion of 7 dehydrocholesterol to previtamin D? Okay, so you need to know this is 7 dehydrocholesterol. So in the presence of sunlight, if you have read pericyclic reaction, so this conrotated electrocyclic ring opening will happen, and in first step it will be ring opening. This bond will break and it will form this free vitamin D3, and then 117 hydrogen shift will occur, will lead to the vitamin D3. Okay, so option B is correct. So in pericyclic reactions, you might have noticed like number of pi electrons will tell you whether it will be con-rotatory or dis-rotatory in presence of light or heat, right? So if some of you have not read the pericyclic reactions, once you read, you'll understand this. Okay, so next question is predict the major product combination for the following reactions. So the reaction is transaminase. Transaminase, what it will do? It will do transamination. So what will be the product here? Transamination means here actually transaminase will have a PLP as a cofactor and then it will lead to this. So PLP, let's see what it will do. If you have a PLP, pyridoxal phosphate will have a aldehyde. First it will have a aldehyde, then it will form this imine. Amine will form imine with aldehyde. Amine will form aldehyde, then base will subtract the proton, and after subtraction of proton, this will go back and subtract one more proton. So you have this kind of, and then water will come, and you'll have a hydrolysis. So you have a transaminated amino acid, and then what it will do? It will again form this alpha ketoglutarate. They have given this alpha ketoglutarate, then it will form imine with alpha ketoglutarate. It will again obstruct the proton, and you'll have this glutamate. So it will show this transamination. So amine and oxygen will be exchanged. So you are getting here this glutamate labeled with 15 N. So this glutamate labeled with 15 N, and you are getting transaminated amino acid and alpha keto acid. So this alpha keto acid labeled with oxycysteine. Okay, so this transamination reaction will occur with the help of PLP, the cofactor. Okay, PLP function of pyridoxal phosphate. PLP is a pyridoxal phosphate. So PLP is required for the enzyme for those reactions like it is required for transamination, decarboxylations, and racemization. So let's see the decarboxylation mechanism. So once you have a PLP and amino acid will form amine, imine, and then base will obstruct the proton from here, and then you it will go for the decarboxylation. After decarboxylation, it will go here, it will go here, and this positive charge will be stabilized. Once you have this lone pair again bound, it will go, yes, and then it will take the proton from here. So now you have this sp3 carbon with the two protons. Then you have a hydrolysis from here. So this is a decarboxylation mechanism facilitated by PLP. Okay, so thanks a lot. This is all for today. We'll meet in next class.